Polymerase chain reaction, or PCR, is a very common technique used in molecular biology to amplify, to generate enormous quantities, copies of a particular segment of DNA that we are interested in. And the way it works is, if we recall the structure of DNA, it typically exists as a double-stranded form. So DNA looks something like this, if I can draw semi-decently. And each one of these strands has a base that corresponds to its other base. So A binds with T and G binds with C and vice versa. And so what happens in polymerase chain reaction is, let's say we have some segment that we are interested in here, and we don't necessarily care about the rest of the DNA extending in either direction. What we do is, step one, heat to melt the double-stranded dsDNA to single-stranded DNA. And so DNA, like chocolate in your hand, will fall apart when we apply heat to it, because these AT and GC base pairs are only held together with hydrogen bonds. And these hydrogen bonds are fairly strong, but when there's a lot of thermal energy present in your system, they will separate. And another side note, GC bonds have 3 hydrogen bonds, whereas AT bonds have only 2 hydrogen bonds. So GC base pairs have higher melting points than AT base pairs. And so after we have arrived at a single strand of DNA that looks a bit like this, the next thing we're going to do is cool solution to 65 degrees C. And when we melt the DNA, this step occurs at approximately 95 degrees C. And so if you're ever in a biology lab, you'll see thermocyclers where people are programming in the unique temperature and duration to hold that temperature at before looping. And so continuing on with step 2, cool solution to 65 degrees, so the DNA primers can anneal. And so what that means is, because we will know a particular region of DNA that we want to amplify, we will have the correct primers that will stick onto these regions in your DNA that we want. And so with all these single strands segments of DNA that exist in your solution at 95 degrees C, when we've cooled it to 65 degrees C, is now cool enough that we can reform these AT and GC base pairs with each other. And so we'll have these little starter primers that are stuck onto the DNA in the regions that we're interested in. And so what happens next in our solution is we heat solution up to 75 degrees C. And again, these temperatures do have some leeway, call it plus or minus 5 degrees C. But at 75 degrees C, there is, so DNA TAQ polymerase can build the nascent strand. And nascent just means growing, the complementary base strand. The nascent strand of DNA. And so at 75 degrees C, in our solution, we will have already present the deoxyribonucleic acids for adenine, thymine, guanosine, and cytosine present. And at 75 degrees C, TAQ polymerase will be able to come in, and on a single strand of DNA, we will have a primer. And we'll recall that DNA polymerase will only work in the 5' to 3' direction. And so these primers, so your single-stranded DNA that we got from melting it would have a 3' direction going this way and a 5' direction going this way. Your primers would have a 5' direction going this way and a 3' direction going that way. And Taq polymerase, I'll just draw it as a block. And also, it's common, Pol will be abbreviated polymerase in MCB. And so Taq polymerase will then latch on, and in solution we'll have D-ATP, D-GTP, D-CTP, and D-TTP. And so Taq polymerase will take in these nucleic acids that are unique to DNA and add them in. And it will continue to do that until it has successfully completed building the complementary strand in the 3' direction. And so once it finishes doing that, the fourth step is to reheat solution to 95 degrees C and repeat steps 2 and 3. And we do this indefinitely until we've either extracted the quantity of DNA that we're interested in, or until we run out of these DNTPs in our solution. And DNTP is a generic term for these 4 nucleotides that I've just written. And so the other important note to realize with polymerase chain reaction is when we perform one cycle, we are doubling the segment number, the number of segments of our interest in our solution every cycle. And so 1 segment becomes 2 segments, becomes 4 segments, becomes 8 segments, becomes 16, 32, 64, 128, 256. And so we have exponential growth in our solution as we continue these cycles. And so after 10 cycles, you'll have an enormous quantity of those DNA regions of interest. And so this concludes where PCR ends. But in terms of molecular and cellular biology, it's just the beginning, because there's a lot of things we can do and interfere with the steps that are pretty cool. And so just to kind of give you guys a taste of it, one of the things we do is, if in our solution we have present magnesium or Mg, so magnesium or manganese ions, what they do is they will interfere with this process. They will interfere with Taq polymerase and cause a much higher error rate when Taq polymerase is trying to find the correct nucleotide to put in. So if Taq sees a G, it should go grab a C. But if we have these ions present in your solution, they will cause Taq polymerase to accidentally grab any other base pair, and this results in us generating random mutations in our complementary strands of DNA. And the reason this is so important is because it is essentially expediting natural evolution, and we're able to use this in lab to discover new proteins and identify proteins and complexes that are very useful to understand in the field of medicine and biochemistry. So I hope you guys found this useful. Let me know if you have any questions, and thanks for watching.